Okay, so the purpose of this video is to take you through what we would have done in a face-to-face lab, or maybe someday I'll use these as intro labs or pre-labs. But what I like to go through here is one of the first things you need to learn is basic heart anatomy. And hopefully most of you have already acquired that. Usually we would have had the convenience of being able to go through things in a diagram, a model, a 3D model, and it's always interesting to me how the brain does not always transfer a diagram to a 3D model. You can find what the tricuspid is on a diagram, but then can't find it on a model. And then also how it can be difficult to transfer from a model to a human specimen. And so we do have plastinated specimens, which are nice because they don't smell so much and they don't really expose you to toxins that might be, we don't know that there's any toxins in the preservative, but you never know. Formaldehyde once was thought to be non-toxic and now it's a type 1 carcinogen. And then we also can move on to a preserved specimen. So I would really take my time with anatomy, maybe take it through all those steps. The way in the online environment you're going to be tested is it's fair to ask you a question about where these are on McGraw-Hill's Revealed product, so their cadaver dissection software. So I could take a picture and show any one of these and then ask you to identify. So what you might do is find a diagram. Web diagrams are fine. Find all these structures just on a picture, and then also go through and in place of the model, find them on the 3D structure that's in McGraw-Hill Revealed. So get your anatomy down. Stuff that's in gray is either anatomically different. It's kind of interesting to look at human specimens because not all of our arteries and veins around the heart look the same. They can be very different. Sometimes you have two anterior interventricular arteries and things like that. So that's why those are grayed. I'm going to take you over here to this diagram, and I actually think I'm going to jump over to a more open space for myself so that I can draw a little bit more. So when I jump over here, you can kind of see that I cheated and I gave myself a little template because I really like Venn diagrams just for organizing information. So I'm going to draw a little Venn diagram already off my lines, but this is good enough. And what I want to point out is there's basically three things that can go wrong with a heart. And we've kind of talked about this in lecture. You can have a problem with blood flow. And so that's going to be heart attack in layman's terms, or a little bit more officially, we would call this myocardial infarction. So ischemia is when there's not enough blood flow, but nothing has died. Infarction is when tissue has died. So over here in the heart, then the next thing is electrical. And I sometimes like to think in metaphors. So like a car needs three things. It needs air, it needs spark, it needs fuel. And your heart needs blood flow, it needs electricity. So if this goes wrong, you have what's called cardiac arrest. Then over here would be problems with the muscle. And so the heart muscle is too thin or it's too thick. And so this is heart failure. Now I really like the heart because it expresses a common theme in this class, which is humans are brilliant because we come up with these great clinical tests, but then we're idiots because we don't do them enough. And I don't want to sound sexist, but it's great that we're so cautious about breast cancer because 1 in 8 females will get breast cancer and 1 in 30 will die of breast cancer. So it's driven this drive to do clinical testing, but we don't have that in the heart, even though that kills about 28% of us. So you should probably have a yearly ECG, especially since that it doesn't even involve radiation, whereas mammography can actually cause problems. But we should do that more often. Or nowadays, you can get your own. You can get a Cardia for $100 at your Walgreens. You can get the Apple Watch. So we're very smart because Einthoven was the guy that came up with EKG. He was really interested in electrical signals in the body, and so he put his hands in buckets of salt water, feet in buckets of salt water, feet in buckets of salt water, and basically measured electrical current with a voltmeter. And he'd already done this, and so he'd already gotten up into O, measuring things. And so he was at P, and he noticed this blip, and he called it a P wave, and then he saw something called a QRS, and then a T, and he realized this is his heart. And for the next 100 years, we've been analyzing EKG and manipulating it and doing it in different ways, and we're still doing this today aggressively. Little things like the newest one I read about was like even the slope of this line can tell you different things about the heart. So we're constantly getting better at this, but we don't do it enough because what's the most common first symptom of heart disease is death. Heart disease kills a lot of us, and we don't do the things that can help us prevent it. And so, like, some of the examples I want to do is, if you worry about enough heart flow to the heart, you can do things like you can look at the T wave in a stress test. Or you can do echocardiograms. We have the means to do these kind of things. CHF could potentially be right in here because you can see the lack of blood flow, or too much blood flow actually can show up on something called a QRS axis. And then you can also see heart failure, so you can see down here CHF in that. Over here, electrical problems, of course, you've got all kinds of things like ST interval, so EKG kind of stuff. You can look for bradycardia. So if you have an Apple Watch, it can tell you that your heart rate is too fast and maybe you have an infection. I suppose down here is any kind of heart failure, and so CHF could be clearly way down here. I put it there because you can tell. Sometimes you can see the effects of CHF on blood flow. The heart muscle gets too thick, the heart muscle gets too thin. But again, the main common thing, and I won't go on too long on this, but this is going to kill. This is the most common signal or symptom of heart disease is death. So we're going to go through, and it's kind of a crude drawing here. But one of the things we would normally do in a face-to-face environment is we would go through, we have a heart with valve disorders, and I kind of want you to be able to say, how would we diagnose this, or what would the symptoms be? So coming up today, we're going to do a case study of valve disorders. So I would like you to be able to, if I show you a heart valve disorder, so the way this would work face-to-face is during the practical, I'd give you a heart valve you'd have to identify it as a heart valve disorder, and then either give symptoms based on the case study you're going to do, or a diagnostic. So we're going to go over EKG. We're going to do things like size of the P wave, QRS axis. You could also measure it with blood pressure variability, and pulse pressure could tell you have a heart disorder. So we've been talking about that a little bit. MI, the symptoms, you would know those. So pain in the left, women usually feel uncomfortable, feel real faint. How would you diagnose it? You can diagnose it with changes in the T wave, ST changes, so that's a stress test, QT interval. So I want you to link up, when we talk about EKG, to a heart pathology. CHF, again, you're going to want to know the symptoms, because CHF, we're going to do a case study on that. That's case study number 2. But I also would like you to diagnose CHF before it becomes a problem. So again, hitting this theme, we can diagnose these things before the person drops dead, not to be cruel. And you can diagnose CHF with changes in the R wave. That's not the best, but changes in something called the QRS. And we'll talk about the QRS axis when we get to EKG. Same thing with a pacemaker. You can diagnose arrhythmias with too fast of a heart, too slow of a heart. And you can also diagnose things like heart block, where the conduction signal gets stuck in the AV node. And it's usually due to, again, a lack of blood flow. So here we are back to, we could put that right here. So AV block would go right here, because it's a problem with electricity, but it's usually a problem with those cells not getting enough blood, and so it's a problem with blood flow too. So this is kind of just a real early kind of look at. I would like you to take our pathologies and be able to divide them up into, is it a blood flow problem? Is it an electrical problem? Is it a muscle problem? And then what could the symptoms be? And what would be a diagnostic that would help this person not suffer death without knowing they have heart disease? Obviously, I put this up in the intro orientation, that there's a lot further you can go with this. Kind of what I put on just now is what I would expect you to know. I would like you to refamiliarize yourself with heart muscle. And also remember that muscle has 4 different distinguishing characteristics between skeletal smooth muscle and cardiac muscle. And I would like you to kind of know why. We did talk about that. I think it was in Thursday's Zoom. I will switch over here though and show it anyway. So cardiac muscle, the first thing about it is it's branched. Then also, I should have We'll see if this works. There also should be these little gaps in here, and those are called intercalated discs. And then these cells abut other cells too, and then they have these like striations. Okay, so the first thing is the nuclei off to the side. Now it differs a little bit, but I would challenge you that if you looked at a cross section of cardiac muscle where there's all these muscle fibers in here, most of them are going to be towards the side. Some are going to be in the middle, but most of them are going to be towards the side. So, and why do you want them off to the side? Because you've got these ions, like calcium, that have to flow from cell to cell. And so you want the nucleus off to the side. So the anatomy is right here. The physiology is to let ions flow. Okay, why are the cells branched? So I guess I could have put shape here. And they're branched. And why are they branched? Because in the heart, electricity has to flow multiple directions and it has to split. So signal has to spread. And then you have over here presence of intercalated disc, and that's only found in heart. And that's to let the ions flow from cell to cell. In your biceps muscle, each muscle fiber is triggered by its own neuron, but there are no neurons going into the heart muscle cells, and so they trigger each other by ions flowing cell to cell. And then the last one is striations, and that's just shared with skeletal muscle, and it's just due to the overlap of actin and myosin. So remind yourself of those. No computer exercises for us. I would like you to go through and find examples. I know that we're not going to be able to look at our specimens, but I would like you to go through and find examples of these. So MI, you're looking for heart attack. And a classic example, and this is going to come back to why we care about blood and why we care about muscle, is so I'll try and draw a fancy heart. Okay, so when it comes to MI, what I would like you to see okay, not so fancy, but what I would like you to see, especially if you look at our heart specimen sometime, is the damage tends to be on the inside of the heart. Why is it on the inside of the heart? Because that's the last place that blood gets to. So blood comes out of the front of the heart, goes around to the back of the heart, moves from out to in. And so the inside of the heart is usually the last to get blood. So you usually see MIs on the inside of the heart. What does CHF looks like? CHF is the one, I'm going to refer you back to lecture, where the heart gets bigger and bigger and bigger because it's stressing against high blood pressure. But then since the blood vessels are on the outside, the problem is the blood doesn't get to the inside. And so you end up with a massive heart, but the walls are very thin. So this is supposed to be the pulmonary trunk coming out of there. And this is supposed to be the aorta coming out of there. And you end up with really thin-walled heart. So that's CHF. I'd also like you to look at our pacemaker heart. And our pacemaker heart, one of the things you'll notice, and we talked about this in lecture when we talked about the Wiggers diagram, is you don't really care about your atria too much. And in fact, you just want to improve flow. And so while your normal pacemaker is here, when they put artificial pacemakers in, they put it down here. They might trigger the atria too, but they don't put it down in the left ventricle. They put it in the right ventricle so that when the heart contracts, it contracts towards the great vessels, and it's easier to get the blood out. So that's kind of cool. And then And then we talked about this in lecture a little bit, but I think I'm going to draw it again. Is this right here is the mitral valve, or bicuspid. Okay, so it's been like 10 years since people took fen-phen, but the problem with fen-phen is it damaged the mitral valve. It seems like a small thing, but it's not a small thing if you understand the purpose of the valve. So first of all, if this pinches because there's stenosis, then that means it's hard for blood to get through here, and it means the pressure is going to build up right there. And then also, since the blood can't get through to here, the pressure is going to go down here. Where's the blood coming from when it goes into the left atrium? Well, it's coming from the lungs. And so this is going to lead to pulmonary hypertension. So the blood pressure is going to go up in the lungs. And here's where I don't want you to be clinically accurate. I just want you to think through it. And you need to know this because it's also the case study. Well, you're going to have increased pressure in your lungs. And that's going to damage vessels. And that's going to lead to embolisms, so clotting. It's also going to lead to fluid in the lungs. And that fluid is going to make it hard to take up O2 because there's fluid where you should have a space to pick up oxygen. So that's pretty bad. That's a pretty bad cluster mess. And then if I come down here to this one, there's not enough blood that's going to go out the aorta, which I'm going to write down here because I have space, but this is going out to the systemic system. And so this is going to lead to systemic hypotension. And here again, you don't have to be clinically accurate. You just have to be logically accurate at this point in your career in A&P. And so you know you're going to have decreased blood flow. And so that's going to lead to things like syncope, fainting. It's going to lead to fatigue. A big thing it's going to lead to that we'll wait till later is it's going to result in decreased kidney filtration because you don't have enough fluid. You don't have enough pressure to get rid of fluid by the kidneys. And then you're just generally going to have decreased nutrient delivery, which again is going to lead to fatigue. So you see, I mean, I just remember when this court case came out and the people that took fen-phen won billions. And this was like 15 years ago when billions was really big. And so it seemed like weird, but it doesn't now because you realize that little valve, if it fails, can create a pretty huge cluster mess. And so that's why I would like you to go through valve replacements. I guess I didn't do bypass. Look at bypass too. Look up some figures of bypass. So you would find them as CABG, CABG, for coronary artery bypass graft. So pacemaker, valve replacement. I forgot to do bypass, but there's not too much to write. CHF and then MI. What I'd like you to do here, and I'd like you to do it relatively soon, because I want to go through EKG with you, and I want to show you all the things that EKG can help you identify. So don't just copy things down from Wikipedia, but look things up a little bit. You don't have to write as much as I did. Just be able to describe what an arrhythmia is. The heart's going too fast, the heart's going too slow. Hypertrophy, when the heart is stretched, it grows. So simple things like that, that's all I require. But again, those are things that will pop up on an EKG. So I would like you to do that before we do EKG. This case study, it's kind of dopey, I know. But basically, back in the day, I used to not tell students what this is. And if you go to Wikipedia and you look up these drugs, which are analgesics, or you also look up fen-phen is in here somewhere, and you also look up rheumatic fever, all of those seem to damage the mitral valve. So this is mitral valve stenosis, and again, you don't have to be clinically accurate, but I'd like you to think it through and come up with some good ideas about what that's going to do. And again, you're going to go back and refer to this diagram to help you with that. And then the extra credit, there's no such thing as extra credit in AP if you're offered points. And in fact, I'm kind of a jerk about this because I want you to do this case study and I'll ask you questions about this. And again, it's to emphasize the point that there's no such thing as extra credit. But also these 10 points of extra credit are going to be helpful in lab, because then you can miss 10 on the practical, or if you don't turn in a lab, then this will make up for it. So this one is CHF. So I'd like you to read through this. So why does he have pitting edema? Why are his nail beds cyanotic? Why does he want to sleep sitting up? Why is he coughing? Why does he have distended neck veins? So CHF, I've been saying this multiple times, it's a great thing to look at early in AP2, because we're going to constantly study the interaction between the heart, the lungs, and the urinary system, and how when those three fail each other, they can bring all three down, and then you're going to bring everything else down too. Okay, so that's the lab. Thank you.